Dear visitor, I'm Lorenzo Muzzi and welcome to the poster presentation entitled Thermogelling Biomimetic Hydrogel Supporting 3D Neuronal Networks. Biomaterials are being utilized to develop biomimetic pro-regenerative construct in the form of 3D scaffold to augment functional neural tissue repair and regeneration. In particular, engineered natural biomaterials able to perform the soul-gel transition in the presence of cells under specific external stimuli have been proposed. The great interest in these stimuli-responsive hydrogels relates in the possibility to encapsulate cells, in their application as ink in 3D bioprinting and in them in vivo injectability for repair or regeneration. In this work, Thermogel was developed as an artificial matrix to encapsulate neuronal cells and to support neural network development for in vitro studies and as injectable hydrogel for the in vivo application or innovative ink. Chitosan is a widely used biomaterial, well known for its good properties. This polysaccharide, in combination with basic compounds such as beta-glycerol phosphate, beta-GP, becomes thermosensitive in diluted acids and can undergo gelation around body temperature. In particular, their temperature-dependent gelation mechanism was found to involve the heat-induced transfer of proton from chitosan to glycerol phosphate, which reduces the repulsive force among positively charged ammonium groups and allows interaction of chitosan chains. Two different formulations were developed and chitosan beta-GP thermogels were prepared at 4 degrees Celsius by mixing sterilized chitosan solution with sterilized beta-GP gelling agent solution added drop by drop. Other hydrogels had a final concentration of 2% chitosan. 3D neuronal cultures were obtained by adding induced neurons mixed with cortical astrocyte in a 1:1 ratio in the chitosan beta-GP solution. Now some of our results. So through the rheological characterization, we obtained that both formulations start the gelation process at 32 degrees Celsius and the complete gelation was observed after 20 minutes for 2.5% of chitosan beta-GP formulation and 5 min for the other formulation. The injectability evaluation was performed varying the dimension of the needles at fixed flow rate at 500 mm/seconds. The experimental result demonstrated that only the 3% chitosan beta-GP formulation was suitable for a correct extrusion, while the 2.5 chitosan beta-GP formulation showed a non-continuous flow and didn't gel during the extrusion process. The mechanical characterization of the gels with and without cells were carried out 1 day after the sample preparation and after 21 days in vitro. In particular, chitosan 3% is positively affected by the presence of the neuronal cells with an increase of the elastic modulus from 7 kPa to 23 kPa. Interesting, as it's possible to observe both thermogels show elastic moduli that fall in the same range of brain tissue. Both thermogels were morphologically characterized by scanning electron microscopy measurement, and both thermogels had an average spore diameter of 20 microns. The immunocytochemistry and confocal microscopic characterization allowed us to gain information on the morphology of the 3D structure of mature neural networks after 35 and 42 days of cultures. Neuronal cells in green and astrocytes in red were homogeneously distributed on the thermogel and they create a very 3D dense networks. Regarding the cell morphology, the neural soma was round, like the ones observed in the brain tissue. The astrocyte-glia fraction also proliferated. Its morphology was again similar to the one found in brain tissue. For the electrophysiological characterization, 1 minute of spontaneous activity of a cultured recording during in vitro 32 was observed in the raster plot. The graph represents with each blue point a single detected spike at a precise electrode in the microelectrode array device. The adjuvant graphs we gathered all the detected actual potential shapes in 2 different active electrodes. Each blue line is a single action potential, with the peak centered at 0. The black dashed line represents the mean action potential shape of the electrodes. The preliminary results showed that both formulations are suitable for neural cell encapsulation, being able to sustain the viability and the growth in order to obtain a very biomimetic system. In particular, the injectability test showed that the 3% chitosan beta-GP formulation is potentially usable for minimally invasive in vivo application and may thus represent an alternative ink for 3D bioprinter. With this, I'm concluding and I would like to thank all the part that were involved during this experiment. Thank you for attention.